Okay. So we'll get into nutrition today. What you put in your mouth, what you chew, swallow, eat, and drink, and whatever. Macronutrients, we're not going to talk about micro anything, just the macros, carbs, fats, proteins. And if you count alcohol as a macro, we're not going to get into that one either. We did alcohol in Stress Phys, and we talked about mTOR there. But just carbs, fats, proteins, we'll talk about that stuff. Oh, you know where to find everything. Just a reminder that the case study is due on Friday, and next Wednesday, not this Wednesday, will be the exam. Hypertrophy, you know, the difference, the genetic expression part of transcription. And so we're thinking steroid hormones, transcription, translation. This is a lot of mTOR activity, is translational efficiency and capacity. And mTOR complex 1 is doing most of that. You know the proteins in that complex. You know the upstream stuff. You know the downstream stuff. You know the interaction between MAPK, mitogen activated protein kinase, and RSK through tuberin and raptor, and then the PI3K pathway going through PKB. This is really the bulk of protein metabolism, really the bulk of muscle regeneration, repair, remodeling, hypertrophy is this PI3K pathway. Secondarily, we have MAPK, and they do converge. At the end, there's a lot of convergence. They interact in the middle. Stuff activates both PI3K and MAPK, and sometimes we get cross activation, sometimes we get cross inhibition. It really depends on context. It depends on how we're measuring, what cells we're looking at. It depends on what animals. Context matters in that stuff. So for the immune chemical, you know this stuff, the immune system, a lot of this stuff is going to overlap. You know, when you're looking at immune and chemical, myokines, stuff like that, is going to overlap with the endocrine system in terms of we have these chemical messengers that are going to relay signals released from stuff. That's the same thing up here and down here, but we've separated them in classic hormones and sort of chemical protein activators and inhibitors of mTOR. And so that was the prostaglandins, that's a lipid, and then a bunch of cytokines, right? The interleukins and interferon gamma and all these myokines. Mechanical tension, remember, there's a lot of mechanosensors, proteins embedded in tissues that will detect loads, load profiles, and the nature of those loads, how heavy you're lifting, how fast you're lifting, the angles, all that stuff, duration of tension, all of that. And the major ones, like alpha-actinin, I mean, there's a lot of these, but the three we talked about mostly are cadherins, titin, and integrins. Remember, integrins are integrating that extracellular matrix to the inside of the cell. They're a transmembrane protein going through focal adhesion kinase. Titin is an intracellular load, meaning within the cell. It goes from the zetas to the M line, huge, long protein. And then cadherins are cell-to-cell communication, and a bunch of different pathways that they're going through. We'll talk more about intracellular calcium a little bit today. Diacylglycerol kinase, remember this is phosphatidic acid. There's a few ways to get phosphatidic acid, but with mechanical loading, DGK is going to be that predominant way to get your phosphatidic acid. Endocrine system, we'll do a little bit more justice to in the rehash, and then we'll get into nutrition. So remember, we have, let's just call these polypeptide hormones, meaning some of them are a single amino acid, an amine hormone, a bunch of stuff made from tyrosine. Really, we have all these amine hormones, catecholamine, stuff like that, T3, T4. But this polypeptide class, some of them are a single amino acid, some of them, there's oxytocin, there's a little string of amino acids in there. Some of them are protein hormones, growth hormone, much bigger, a long string of amino acids. But all of these are water-soluble, fast-acting, shorter lasting, but pretty rapid effects with these protein and polypeptide, amine hormones, things that are basically proteins of varying lengths. And then we have our steroid hormones. Remember, steroid hormones are made from cholesterol. We make steroid hormones out of cholesterol. And then eicosanoids. I put the eicosanoids in that immune chemical section, but these are lipids. They're made from 20 carbon fats, mostly, almost entirely arachidonic acid. You know, that 20 carbon lipid in that phospholipid bilayer, 10 or 20% of it in there, get into a skeletal muscle cell. And in that phospholipid bilayer, you have a bunch of arachidonic acid. Liberate that arachidonic acid and convert it into stuff. Those are eicosanoids, but those have signaling properties in the exact same way that, you know, these hormones, these polypeptide hormones would be doing, cell signaling cascades. And remember the hypothalamic pituitary fill in the blank axis, these axes that we have in the anterior pituitary. So the hypothalamus, remember, that's a brain region, that's not a gland. The hypothalamus is a little hunk of brain tissue, and that regulates a lot of these bodily functions. And it communicates directly with the pituitary. Now, through the anterior pituitary, it's a capillary network. It doesn't go into systemic circulation. It's a direct circulation from the pituitary, from the hypothalamus down to the pituitary. And the hypothalamus tells the pituitary what to make. The anterior pituitary makes other stuff, sends it out, and we have these signaling cascades. In the back, at the posterior pituitary, that's connected through nerves. The posterior pituitary isn't actually making its hormones, you know, vasopressin. That's made in the hypothalamus, but stored in and released from the posterior pituitary, oxytocin also. So the posterior pituitary isn't as much about signaling cascades, about axes rather. The anterior pituitary, that's these axes really going through the anterior pituitary. And again, hypothalamic pituitary, and you end somewhere. Now, if you're going to end in the adrenal cortex, you're going to get cortisol, right, from the adrenal cortex. If you're going to end at the gonads, right, this is the hypothalamic pituitary gonadal axis going through luteinizing hormone through the pituitary for testosterone. Liver, get your IGF. Thyroid, we'll talk a little bit more about the thyroid today, but T3, T4 is what we call them. T4, thyroxine, you make a lot of that. The biologically potent one is T3, tritothyronine, but we'll talk a little more about the thyroid as we get into this stuff. But remember, for steroid hormones, their major effect, the primary effect of these steroid hormones is going to be transcription, right? It gets into the nucleus, binds to a little patch of DNA and regulates transcription. This is the primary steroidal effects. They don't bind to cell surface receptors for the primary effects. It's inside of the cell, and then for the androgen receptor, you're going to bind to a little patch of DNA. So, but remember, they're all from cholesterol. All of these steroid hormones are coming from cholesterol. Cholesterol in those is modified a little bit. So the androgens, dehydroepiandrosterone, that's very weak. That one's legal. You can go buy that one. This one became legal in the Mark McGuire era, androstenedione. And then testosterone, dihydrotestosterone, that's the one where, like, prostates get huge. So if people are doing excessive amounts of anabolic androgenic steroids, you'll often see conversion to estradiol and to dihydrotestosterone. So this is the big prostate, and this is big breast tissue, gynecomastia. So aromatase would be the enzyme that would do this conversion. So people would sometimes do an aromatase inhibitor or whatever. But this is what you want for the androgens. It's a hypothalamic pituitary, go through luteinizing hormone, gonadal axis to get your androgens. And most of it is coming from the testes. You get a little bit from the adrenal cortex. Corticosteroids, mineralocorticoid, glucocorticoid, right, aldosterone and cortisol. You also get a little bit of androgens, a very small amount out of the adrenal cortex. The cortex is where you make steroid hormones. The adrenal medulla, you're getting some amine hormones out of there. But the primary effect of testosterone, that's the steroidal part. Let's bind to the androgen receptor and initiate the transcription component of it. But also, remember with testosterone, we're getting more expression of IGF-1. That's how growth hormone does most of its tissue interaction. Most skeletal muscle tissue remodeling and hypertrophy is through IGF-1. Testosterone also works a little bit through IGF-1. And you see selective hypertrophy in some of these studies at the higher threshold motor units. And so P70S6K takes a little while, right? Takes a little while with testosterone to get this mTOR, the mTOR signaling. Immediately downstream from mTOR, one of those targets is P70S6K, and it takes a little while to get that going. But with the MAPK signaling, it's like 5 minutes. 5 minutes, and you see MAPK signaling. And we know a lot of those calcium kinetics are going to stimulate. If you can get some intracellular calcium, you can stimulate MAPK signaling pretty effectively. And it goes up and it goes down. It goes down around the same time. Remember this cross inhibition. So as mTOR is roaring, MAPK is backing off a little bit. Contextual. It's contextual. But again, we're getting MAPK, we're getting PKB, that intracellular calcium release is particularly promotional to the MAPK signaling. So that intracellular calcium, calcineurin signaling is helpful. Also, though, testosterone is inhibiting LKB1, liver kinase B1, and again, don't think of it as living in the liver. It's all over the place. There's a lot of literature about it in the liver, but this is ubiquitous. And LKB1 is this primary activator of AMPK as long as AMP is bound to it. Once you bind AMP to AMPK, we'll talk about that next lecture, that permits LKB1's activation. Once AMPK is on, mTOR is off. And so testosterone is inhibiting that. Estrogen is stimulating. So estrogen is a little bit weird. And so this is just a look at the LKB1 expression. And testosterone, this is a androgen receptor inhibitor. And if you introduce testosterone, LKB1 down, you sort of abolish that. But introduce testosterone and a receptor antagonist, and you see LKB1 elevated. Same thing with DHT, dihydrotestosterone. Remember, that's the one right after testosterone. If you introduce that, LKB1 is down. Introduce that and the androgen receptor inhibitor, LKB1 is elevated. So LKB1 is another mechanism through which testosterone is enhancing PKB, PI3K, PKB, mTOR activation. Now remember with things like World's Strongest Man, where people are doing, or any powerlifting competition, leanness is not helpful. Leanness corresponds to weakness, and fat isn't contributing to the contraction. You know, there's no actin and myosin doing cross-bridge cycling in, abdominal fat, right? However, people who cut a bunch of fat and even preserve all of that muscle are still weaker. The performance suffers. And so in bodybuilding, watch somebody who is stage ready, 3% body fat or something, up on stage. And then look at them when they're off stage. And if you can manage to keep the muscle mass the same, it's sort of impossible, but if you can keep the muscle mass the same, they will be much weaker when they're leaner. And part of that's going to be calcium kinetics. The testosterone and estrogen permitting calcium flux. Part of that strength capacity, part of that performance is going to be through the ability to regulate calcium. And so getting into estrogen, remember genomic effects of estrogen. It's a steroid hormone. That's its primary action, is genomic. But there are secondary non-genomic effects going through mTOR. And one of those mechanisms is the deactivation of tuberin, tuberous sclerosis complex 2, which is what is breaking down that GTP on Rheb, preventing Rheb, RHEB, from activating mTOR. So we'll talk a little bit more about that interaction between Rheb and mTOR today as we get into what protein does to activate it. But estrogen is going to deactivate tuberin. But part of this, again, I refer to it as the deptor of steroid hormones because it has pros and cons for anabolism. There are pros and cons for anabolism. And it's going to turn on, it's going to inhibit tuberin, which is going to promote mTOR, but it also promotes LKB1 and AMPK. Right? And these things, direct and indirect activation of AMPK, that's an mTOR inhibitor. So contexts, it's a bit of a deptor in its relationship. And when you see the effect of estrogen on performance, it gets even more interesting. I'll just read this. So it's clear that estrogen has a dramatic effect on musculoskeletal function in terms of regulating protein turnover. In the past, much of this research is focused on the strong connection between estrogen and bone. However, other musculoskeletal tissues, such as muscle, tendon, and ligaments, have become the focus of more research. These studies make it clear that estrogen improves muscle proteostasis, so that protein turnover in a very healthful way estrogen is doing. Look what happens to muscle mass for postmenopausal women. Look what happens to the protein turnover. It's not good. So estrogen is very good for this stuff. And it increases collagen content, sinew, muscle, whatever, collagen content. However, the benefits on bone and muscle come at the cost of decreased connective tissue stiffness. Evolutionarily, this makes sense since laxer joints and better repair following injury would facilitate healthy childbirth and recovery. However, as more women participate in sports, it is clear that the physiological effects of estrogen contribute to decreases in power and performance and make women more prone to catastrophic ligament injury. Pros and cons. Estrogen has pros and cons. Now, testosterone has pros and cons too in terms of like heart disease. But in terms of skeletal muscle growth and performance, anabolism, testosterone is a kind of a single-faceted character, whereas estrogen plays many roles. Insulin and IGF-1, you know how these things work. It's PI3K, PKB. That's the pathway for these. Now, you will also get some parallel, get some IGF over here, you'll see parallel MAPK. Not as much, that's not as potent. The most potent way that IGF is carrying out its effects is through PI3K, PKB, mTOR. Growth hormone, the major way it's carrying out its effects is this. It's through IGF, right? So you get this hypothalamic pituitary liver axis, and you get growth hormone releasing hormone is released from the hypothalamus, tells the pituitary, the anterior pituitary, to release growth hormone. And then later, you're going to get your IGF, and IGF is going to do most of this bulk of the tissue remodeling and the hypertrophy. It's not growth hormone by itself. Remember, during fasting, you're going to get elevated growth hormone, and you are definitely not growing despite elevated growth hormone owing to the suppression of IGF. IGF is really the major hormonal mechanism for skeletal muscle repair and hypertrophy, not growth hormone itself. But growth hormone does have this JAK-STAT signaling and parallel MAPK and PI3K. There's a little bit of that as well in growth hormone. The major way that growth hormone is going to enact its effect, well, IGF, but through PI3K. PI3K, PKB, mTOR, that's the major way. Now, thyroid hormones, there are studies on both T3, triiodothyronine, and T4, thyroxine. And this one is T3, the more potent of them. And looking at this T3 treatment and the time course when you see this effect, so 7 minutes, 15 minutes, okay, we're getting a bunch of AKT, PKB is AKT signaling. So it takes a little bit of time to get that big signal. But if you compare that to IGF, right, here's your T3, yeah, that's good. Here's nothing, right? We're getting a pretty good signal from T3. It's an even bigger signal from IGF. So thyroid hormones are not as potent as IGF, but they are potent stimulators of PI3K, PKB. This is triiodothyronine over here. There's another one made from tyrosine, the amino acid tyrosine. Epinephrine made from tyrosine. But epinephrine, so this one and this one are both amine hormones, meaning made from an amino acid, right? You don't have like a string of amino acids in there. It's not a protein. Testosterone, you see the cholesterol in there. Now remember that epinephrine has to bind to a cell surface receptor. It has to bind to a cell surface receptor to send its message into the cell. Testosterone just goes into the cell. Yeah, there's some cell surface receptors, but goes into the cell. The androgen receptors are in there. Triiodothyronine, T3, T4, right? Your thyroid hormones, they get escorted into the cell. So it's sort of a hybrid of these two. Structurally, it's this. Structurally, it's an amine hormone. There's no cholesterol over there. Structurally, it's this. It doesn't just cross the membrane on its own, though. It needs transporters to get across. And its effect, MyoD, this is for transcription. And this myoblastic termination factor, it's a transcription factor. We talked about this with satellite cells, and when you see the effect of blocking its, the transporters that would get the, in this case, I think they're looking at maybe T4. So this is like they aren't blocking any of those transporters to take the thyroid hormone across the membrane. This is blocking one of them, and you get a little bit less MyoD. This is blocking a different one, you get a little bit less MyoD. Block them both, and you get a compounded effect. So if you knock this stuff out, if you prevent thyroid hormones from getting into the cell, crossing that cell membrane, muscle doesn't recover, muscle doesn't regenerate after injury or after exercise insult to the tissue. It regenerates really poorly. So this is part of a very comprehensive hormonal recuperation profile, sort of the cocktail. This one's T4, look at T4 here. And the effect on, this is a phosphorylation site of PKB. There's S6K or P70S6K. So looking at a placebo as compared to T4, thyroxine, which isn't as powerful as T3. But looking at this impact on those. Same thing over here, but with rapamycin. So this is P70S6K right here. So here's nothing. There's a placebo. If you just introduce rapamycin, you're going to knock out P70S6K. If you introduce T4, you get a lot more P70S6K phosphorylation, activation of P70S6K. If you do T4 and rapamycin, it's the same thing as just doing rapamycin. So the pathway through which the thyroid hormones do a lot of their signaling is going to be through PI3K, PKB, and the corresponding hypertrophy. Endocrine system in general, the summary of the endocrine system, insulin, how that works. Really what we're concerned with insulin is carbohydrate intake. Get carbs out of the bloodstream and into the cell. GLUT4 translocation. But additionally, we're going to get mTOR signaling. We'll get to PI3K, PKB, and other stuff gets phosphorylated too. Thyroid hormones, this is PI3K, human growth hormone and insulin-like growth factor, mostly going through PI3K, which is mostly going through IGF. Testosterone, there are secondary effects. Estrogen, there are secondary effects, and these are going largely through PI3K, PKB, as well as MAPK, mitogen-activated protein kinase. Okay, so that's everything that we've talked about so far.
Can we go back for a second?
Yeah. So those are the major ones. There's other stuff, but these are the major players.
Is this going to be added to the review slide?
I'll add it. Yeah, I'll add it to the review slides. Other questions on the content? Am I still on the way? Okay. So getting into the nutritional part of what you put in your mouth. Carbs, fats, and proteins. All three of these matter. We'll spend the bulk of the day talking about protein. But carbs and fats matter too. We'll talk about fats first. Now fats, lipids, right? Phosphatidic acid, that's a lipid. And through mechanical loading, diacylglycerol kinase is the primary pathway. There's a few pathways where you can get phosphatidic acid, but the primary pathway for go to the gym, do mechanical loading, and get phosphatidic acid is through diacylglycerol kinase, using a diacylglycerol as a substrate, phosphorylated, and that's a lipid, fine. Prostaglandins, MAPK signaling, also, right, that's a 20 carbon lipid. So, but you're not eating these things. So we'll talk about supplements. Phosphatidic acid is a supplement. Arachidonic acid is a supplement also. But there's evidence for phosphatidic acid as an effective supplement. You convert it to lysophosphatidic acid, and you seem to get MAPK signaling. There seems to be other effects of how it's working, as opposed to the classic mechanical loading pathway. But eating fat, eating cholesterol, getting a high-fat diet, this is a way to increase androgens. Plenty of work has been done. This is just one study looking at steroid hormone synthesis and the effects that diet has on levels. Now, I've referenced a couple of times, it's something I used to talk about, I've referenced a couple of times. If you eat oatmeal, oatmeal always has this little heart on it somewhere. There's always advertising on oatmeal, heart healthy, contributes to, reduces cholesterol, contributes to a healthy lipid profile, whatever. And beta-glucans are a type of soluble fiber in oatmeal. There are other fibers that do this, but the oatmeal fiber, beta-glucans, that's the famous one that puts the heart on every single package of oatmeal. It binds to bile. This fiber does. Wherever fiber fiber goes to the toilet, right? But if you eat fiber, it's just the tube that goes from like your mouth to your butthole. There's a little tube, you absorb some stuff, you don't absorb other stuff. The stuff that doesn't get absorbed goes in the toilet, and fiber is going to the toilet. Ignoring, if we were to talk about, the microbiome and whatever, some sort of destinations for that fiber that would be otherwise. But fiber goes to the toilet, and if it's bound to bile, bile goes to the toilet too. Usually you recycle your bile. You reuse that bile. You don't use it once and then that's it for bile. Bile just gets used over and over and over. You just recycle it. If you shit it out, you have to make more bile. You make more bile in part out of cholesterol. So you lower your cholesterol that way. So that's the reason that's one of the reasons we're seeing this heart on the oatmeal. And when you see people who need a lower LDL, total cholesterol and LDL profile, because maybe they're having skeletal muscle infarctions, or Gregory House, they're at risk of myocardial infarction, pulmonary embolism, whatever it is. If we're looking at we need to reduce cholesterol, oatmeal is a reasonable way, and go ahead and go with a low-fat, low-cholesterol diet at the same time. That works. However, there's an effect on androgens. There's a simultaneous effect on androgens because those are made from cholesterol. So there's a study here that there are 41 guys, 41 men, age 50 to 63. You don't need to write down the details of the study, but just be aware of what the study finds, and this is typical for results that you would expect from studies like this. Fat and fiber content. The researchers control this in these people. 39 people completed the study. So there are some people who just went baseline to low fat, high fiber. And there was a bidirectional study where some people went from baseline to low fat and then to high fat. What happens to cholesterol and what happens to androgens? And then some people went from baseline to high fat. Let's see if we can drive up these androgens, and then into low fat. What happens after getting an elevation in androgens? And this is what you see going from baseline. So there's the total cholesterol, and they go on this low-fat, high-fiber diet, and total cholesterol goes down. Yay, right? LDL goes down. Yay! HDL goes down too. Not yay. HDL goes down too. Look at the androgen profile though. Here's testosterone. Testosterone goes down. Free testosterone goes down. Dihydrotestosterone goes down. DHEA goes down. You expect all these things. If you're seeing, your total testosterone going down, all the precursors are going to be going down too. Estradiol is down a little bit. Sex hormone binding globulin, that's a binding protein for estrogen and testosterone, androgens and estrogens. So all of this stuff went down. So for guys especially, males and females, but in particular for males, for whom testosterone is variable and very important, a high-fiber, low-fat diet is going to reduce testosterone levels. Testosterone, again, has a primary effects, but secondarily, through mTOR, you will see compromised recovery in diets like this. Now, when people go from baseline to low-fat to high-fat, just look at the total testosterone. All the other stuff has a similar profile, but total testosterone from 15.2 down to 13.4. That's a pretty big drop from the baseline to the low fat. Go into the high fat diet. Into the high fat diet, going back to 13.6. At baseline, they're at 15.2. Go in the low fat diet and you tank your testosterone, it's hard to recover. So even thinking about things like these phasic diets, sort of yo-yo diets, where, all right, I'm gonna, my brief cut phase and then my mass phase and brief cut phase. The recovery might take a little bit of time in androgens. And when you look at the baseline to high fat to low fat, baseline 15, go to 15.8, you can't increase it from baseline. Testosterone, you can't make that go up. Just eat a bunch of bacon. Cut out some fiber. Testosterone will go up. I'm not saying do that because there are risks, right? 15, 15.8, and then down to 13.7, below what baseline was. So both of these end around the same place, 13.6, 13.7. Recovery is a little bit harder for testosterone. And so consider that, guys in particular, but consider that with nutrition. And when people are like, oh, I'm going to try to get lean, I'll go on a low-fat diet. Maybe, depends on the total calories and the exercise profiles, but there are consequences to doing that.
Can you just go back to the previous?
Yeah. Can your eyes see those numbers from there?
Yeah, no, I think I had a question.
Okay.
Oh, yes, that was a question. What was like the time interval between them switching?
Oh, that's a really good question. It was, I don't have it here. It's been a while since I read this study. Really good question. This study is in the PDF bank, and so you can pull that up and see what the time interval. Now, you can recover. But how long does it take to recover is essentially the question of how long did it take to deplete, how long did it take to not quite recover, and how long might it take to fully recover? That last question, this study doesn't answer. But the time course of these effects is in the study. I don't recall the duration, though, of these. All right, moving into protein. Now, protein is just a bunch of amino acids. Remember, you can't take growth hormone orally. You can't take insulin orally. That doesn't. You can't just like take a growth hormone pill. That doesn't work because it's just protein. You're better off eating an egg, right? Or meat or whatever. These things are protein, and proteins are a bunch of amino acids. And amino acids don't all behave the same. Not all amino acids are equal in terms of anabolism. There's sort of a hierarchy of importance of amino acids. But Growth factors, think like IGF, myokine, stuff like that. Mechanoreceptor over here, phosphatidic acid, that's one way to activate mTOR. Growth factors, PI3K, PKB, stuff like that. Nutrients though, amino acids and glucose. Now amino acids, we see an inhibition of AMPK. We'll talk about that, we'll get there. And especially we'll talk about that even more in the next lecture. But amino acids, calcium, calmodulin, and then this regulator. The rags are how we get mTOR to the work site. You can think of the rags like Uber. The rags are the ones that take the mTOR complex to the work site. mTOR doesn't work from home. It needs to be somewhere. That somewhere is mostly the lysosome. It needs to be somewhere specific to get its work done. And rags are the taxis, they're the Uber, the Lyft, or whatever, to get it there. So again, a couple of different pathways. VPS34, this is a kind of PI3K. You can kind of ignore this one, and we'll focus on rags, because that's the primary pathway through which protein activates mTOR, is through rags. It's through translocating mTOR to, in particular, the lysosomal surface. So there's a regulator-rag complex. It's required. You can't make mTOR work with just IGF. If you inject IGF and you are amino acid deprived, protein amino acid deprived, and you inject IGF, that IGF isn't going to work. You need both of these things. You need both of them. But the rags have to be GTP bound to do it, and they will directly interact with a raptor. So the rag complex binds to raptor and then helps it to sort of rotate and attach to that lysosomal surface, and that's where you can activate it.
IGF needs this?
IGF doesn't work on its own. Yeah. You need protein. Now, at the end, I'll talk about sort of conditionally, you can, if you're breaking down other proteins in the lysosome, this is why the lysosome is so important, is you can break down other proteins there and have this sort of amino acid pool available. And at least arginine can be sensed inside of the lysosome, and lysosomal arginine can help get mTOR to that lysosome. So it's not that you need, in every single situation ever, you need to be eating proteins, but you sort of do. You sort of do. Pardon?
IGF-1 and IGF are the same.
Yeah, IGF-1. What IGF-2, this is like for babies. For adults, for tissue remodeling in adults, IGF-1. This isn't like a developmental thing. So this is for everyone in the room, IGF-1. This is the IGF that is this primary tissue maintainer, primary hypertrophy signal, this massive regulator of skeletal muscle. So we need GTP-bound rag to do it. And eating amino acids increases the amount of GTP-bound rag. Eat those amino acids. Now, you'll see regulator commonly. This is, I believe, the article that coined it from 2010. David Sabatini was the senior author. He's probably the best researcher in all of mTOR. Is David Sabatini, and he was the senior author on this paper. And so I think he was the one who coined regulator. But just talking about these different proteins that regulate rags and the mTOR complex, regulator is in rag regulator. So if you see regulator, that's a complex of a few proteins. Now again, lysosomes, this is the major work site. Think about Pacific. Stockton. Like if you ask anybody, where is Pacific? They'll say Stockton, right? Technically, we have satellite campuses, but ask somebody, where's Pacific? Nobody's gonna say, oh, it's in Sacramento. I think that's a law school that has like 1.5 students or something. Stockton is where Pacific is. And to that same extent, the lysosome is where mTOR has to go. And lysosomes can be loaded with amino acids, and you can get intralysosomal amino acid sensing. And not all of them. You're not going to be able to sense all of those amino acids inside, but there's at least one pathway so far that's been outlined. So this is what it looks like, where you have this, again, regulator. There's a few proteins in there. But you have to have GTP-loaded rag. And that binds to Raptor. Once you get that bound to Raptor, you're able to adhere to the lysosomal surface. You're able to get to the work site. There's no Zoom lectures for mTOR. It has to actually show up to the classroom to do its translation. It has to get, again, primarily to the lysosome to have its Reb interaction. It can't interact with Reb out in the cytosol, wherever it lives, wherever it's just hanging out. It cannot interact with Reb. Reb is at the lysosomal surface. The last 15 amino acids in Reb help it adhere to the lysosome. So you see Reb right there. Reb at the lysosome. So in that string of amino acids, that little end of Reb helps it adhere to the lysosome. And that's to get that Reb interaction with mTOR, which is necessary to turn on mTOR. mTOR has to be here. So that's the reason IGF on its own is insufficient to activate mTOR, because it can't bind to Reb. It's not in proximity to Reb to turn on mTOR. So the amino acids, what amino acids are doing this mTOR translocation? What amino acids specifically are the Ubers and taxis and lifts of mTOR to deliver it where it's supposed to go? Originally, the work was on just eat the whole protein. Just eat all the amino acids. There's 20 of them, put them all in your mouth. And what people found was the essential amino acids, 9 essential amino acids, those ones tended to be more potent. You could remove the non-essential amino acids and still see basically the same signal with mTOR. And then people started looking at, like, well, among the essential amino acids, there's some branched chain amino acids, leucine, isoleucine, valine. There are branched chain amino acids. What if we just looked at those ones? And they got most of mTOR signaling through those branched chain amino acids. And then there's a little leucine. Just kind of narrow it down to leucine, and you're seeing a great signaler with leucine. The most potent mTOR signaler is leucine, but it's not the only amino acid that mTOR is sensitive to. It just is the most important one. If you were to do a supplement, and you were to make your own supplement, and you're looking at the perfect protein composition for post-workout, or take it pre-workout, or that's fine too. Take it around your workout. A peri-workout protein supplement. Leucine, that's your number 1. Arginine, that's your number 2. It's a conditionally essential amino acid. It's not actually an essential amino acid, but arginine number 2. So leucine number 1, arginine number 2, and lysine and methionine. These ones, one of them is going to be a bronze medalist there. Glutamine is also has an effect. But a lot of amino acids are recognized by the cell. It doesn't seem like all of them are. Now this work here, so David Sabatini's lab, phosphorylation of P70S6K, remember immediately downstream from mTOR. Phosphorylation state of this means mTOR is on. Introduce rapamycin and that's off. So all 20 amino acids, pretty potent signal. Just leucine, lysine, and arginine, 90%. 90% of mTOR activity with just those 3. Now think about evolution could not anticipate the invention of hypodermic needles and chemistry labs, of modern science. Historically, people ate the whole protein. You eat the whole animal, right? You don't isolate the individual amino acids from the chicken thigh or whatever. And so you don't actually need, evolutionarily, a cell doesn't need to know that every single amino acid is there. It can just have a couple of representatives. If I'm like having class and I'm like, oh shoot, was there class today? Is it a holiday? I don't need to see every single person show up. I just need a couple of representatives. Like, all right, Cameron and Daniel here. All right, there must be class. Yeah, I just need a couple of representatives. I don't need every single person in class to know whether class is happening. And you don't need every single amino acid to be present to know whether protein was ingested. The cell recognizes a few as representatives of the whole protein and translocates mTOR accordingly.
Now, is this, when you're eating these amino acids, it's ingested and like orally?
Yeah, orally. Yeah, ingested. Now you can break down proteins, right? Ubiquitin proteasome system, the lysosome, break down proteins, and you can sense amino acids. You know, especially intralysosomal arginine sensor. But orally is the primary signal. Letting your body know, and I'll talk about this at the very end, letting your body know, hey, there are amino acids on board. We can actually make stuff. The resources are available. Let's use those resources. If the resources aren't available, what are you supposed to make proteins out of? The proteins in your body. You have to break down, muscle, break down proteins in your body to make new proteins. mTOR is unlikely to be activated, certainly is not going to be hypertrophically activated if it doesn't sense an abundance of amino acids. It's not going to start linking together amino acids with the resources it doesn't have. You can make it do that. It's a little bit harder. So combinations of particular amino acids can signal mTOR pretty much fully. You don't need all of them. Now with arginine, this is that intralysosomal sensor, SLC38A9. It's like the way stars are named, which is terrible names for some of these things. But I'm unaware of another amino acid sensor inside of the lysosome. There's probably something in there. Doesn't seem like you can sense leucine inside of the lysosome though. Cestrin-2 is your leucine sensor, and that's out in the cytosol. You have a cytosol arginine sensor as well. But arginine is sensed both in the cytoplasm and in the lysosome. Lysosomes don't account for that much of a cell, 2% or something like that. They don't account for that much of a cell. But they're critically important to hypertrophy. So, regulating the RAG-induced translocation of mTOR to the lysosomal surface, you can sense arginine within the lysosome, degrade a protein, free up some arginine, you can sense that, and that's one way that you can help relocate mTOR to the work site. So here is the lysosome, right? This green thing down here. Inside the lysosome, you're degrading cells and stuff, or you're degrading proteins. There's that SLC38A9, there's that intralysosomal arginine sensor. Up here though, CASTOR, C-A-S-T-O-R, it just stands for cellular arginine sensor for TOR. CASTOR, cellular arginine sensor for TOR. We're getting into some quadruple negatives here. So if you thought PKB inhibits the thing that's inhibiting the thing that turns on mTOR was bad, right? PKB turns off tuberin. Tuberin was inhibiting Reb. Reb turns on mTOR. This double negative makes it positive. We're getting into quadruple negatives to make a positive now. GATOR1. GATOR1 is this primary inhibitor of this translocation of the RAG complex. Gator-1 is this primary inhibitor of the RAG complex. Gator-2 is a promoter of mTOR because it inhibits the thing that's inhibiting the promoter. So Gator-1 inhibits RAGs. RAGs are necessary for mTOR to be at the work site. Gator-1 is an inhibitor. Gator-2 is an inhibitor of the inhibitor. Let's start with castor. Castor, that cellular arginine sensor of TOR, is an inhibitor of Gator-2, which is an inhibitor of Gator-1, which is an inhibitor of the RAG complex. Arginine inhibits that. So arginine is an inhibitor of the inhibitor of the inhibitor of the inhibitor. So it's a promoter. So arginine is a promoter, right? So it's a little bit easier for the intralysosomal one. Cestrin-2, this is leucine is going to inhibit, it's the exact same thing as castor, but it's leucine, right? Now leucine, there's a little pocket where leucine binds in cestrin-2, and that prevents it from inhibiting Gator-2. And then so Gator-2 can then inhibit Gator-1 and stop Gator-1 from inhibiting the RAG complex.
What was Gator-2? Was a promoter of?
Gator-2 is Gator-2 is anabolic. It's a promoter of RAG-bound, mTOR, getting that RAG complex to bind to raptor and adhere mTOR to the lysosome. Gator-2 is a promoter by inhibiting Gator-1. So Gator-2 inhibits Gator-1. Gator-1 is inhibitory. Gator-1 is kind of like tuberin, in a way. Gator-2 is inhibiting that. So this is, think of this as like PKB and this is tuberin. But cestrin-2 and castor-1, those are both inhibiting Gator-2 until you get the appropriate amino acids in. Now the appropriate amino acids, technically, in this little slot, the little pocket where leucine binds, if you really get a bunch of isoleucine or methionine, if you get a bunch of those, you can cram them into that pocket, and that'll sort of work. Not well, though. Leucine is really what binds with ease there. That pocket is set up specifically for leucine to bind. It's possible to just overload, a bunch of isoleucine, whatever, and cram it in there, and fine. Yeah, but that's not really a practical way of inhibiting cestrin-2. Leucine binds, gets in this little pocket, which inhibits cestrin-2's ability to inhibit Gator-2. So Gator-2 is turned on. If you can inhibit this, you turn on this. If you turn on this, you turn off this. If you turn off that, you allow mTOR the freedom to work. Okay, does that make sense? Quadruple negatives are not intuitive. And then Kickstart, right? This is like pH of a cell, right? Here's protons, is pH of a cell. Ignore this. And then Kickstart is what recruits Gator-1 to the complex. But you can ignore Kickstart too. And this is for pH stuff. You see the TSC complex, tuberin, red. Let's break down the little GTP on red, and you need GTP bound red to activate mTOR. But that only happens if mTOR is here. mTOR cannot interact with red wherever. It can't just be anywhere in the cell and interact with red.
Tuberin changes GTP to GDP?
Yes. Yeah, tuberin is what facilitates this GTP to GDP. And you need GTP bound red to activate mTOR. Just as you need GTP bound RAGs. Now leucine gets into the cell, LAT1, you don't need to know this, but there's a transporter to get leucine into the cell. So this is another look at the same thing. There's cestrin-2, that's the leucine one. leucine binds, there's a little pocket, leucine binds to cestrin 2. That turns cestrin 2 off. It prevents cestrin 2 from inhibiting GATOR2, allowing GATOR2 to be turned on. And on GATOR2 inhibits GATOR1. GATOR1 was inhibiting mTOR. RAGs, inhibiting the RAGs, and inhibiting the activation of mTOR via location. Now here's leucine coming out of a lysosome. I am unfamiliar, and all the work I have read has never identified an intralysosomal leucine detector. It's only that cytosolic leucine. Eat leucine, though, when you get it into the cell. Eat leucine, LAT1 gets it into the cell, and cestrin 2 is the sensor. Now you will sense nutrient availability, and you will sense nutrient scarcity. Nutrient scarcity, these things are really responsive to. They will be the cestrin 2 and castor 1, the cellular arginine sensor. Those are really sensitive to scarcity of those nutrients. And inhibiting the localization of mTOR to predominantly the lysosomal surface. Now it gets more complicated. That M of SAM, that's methionine. So there's SAMTOR. We'll talk about that one briefly. It seems to be a little bit effective. There's leucine, there's cestrin 2, there's castor. But out here, there are other places where mTOR is capable of working. This is the lysosome. This is the major place where mTOR can work. Like nucleus, there are sites where mTOR can work. Again, Pacific has satellite campuses. mTOR has satellite work sites. But really, the lysosome is the only one that's actually really studied because the bulk of mTOR, of protein synthesis is happening there. It's also an amino acid pool inside of the lysosome. So, although the lysosomal surface has emerged as the main cellular location for mTOR complex 1 activation by amino acids, additional compartments, roughly half of mTOR does not co-localize with lysosomes. That doesn't mean half of protein synthesis, but the location of mTOR. The best work site for it is the lysosome. But there are other work sites. You don't need to know any of them. None of them are really effectively studied. The lysosome is the one that's well understood, pretty well understood. Red, ROS homolog enriched in brain, red, right, the thing that activates GTP-bound red, the thing that activates mTOR. You can find that elsewhere. Peroxisomes and Golgi, endoplasmic reticulum, it lives elsewhere. Tuberin can be found elsewhere also. Stuff that regulates mTOR is not exclusively at the lysosome. mTOR can work on other surfaces as well, but the lysosome is the primary one. Now, methionine sensing, this is that SAMTOR one. So SAMTOR is what it binds to. The M is the methionine. And releasing interaction with GATOR1, if we come back here, you can see it better. So there's, I think, methionine inhibiting SAMTOR, SAMTOR turning on GATOR1. So if we inhibit the thing that's promoting the thing that inhibits amino acid signaling. GATOR1 is your primary inhibitor of amino acid signaling.
SAM inhibits SAMTOR?
Yeah, so think methionine, a little bit modified. Methionine, the amino acid, inhibits SAMTOR. SAMTOR is promoting GATOR1. GATOR1 is the primary inhibitor of amino acid activation.
By the rags, right?
Pardon?
By the rags.
By the rags, yeah. By rag-induced localization of mTOR. So methionine inhibits a promoter of GATOR1.
So it makes mTOR turn on?
it helps mTOR to turn on. Yeah, but again, not all amino acids are similar in their effect. Leucine is the most important, arginine is the second most important. But also methionine works, and there's the pathway in which it works. You know, not all Pacific departments have as many students. You know, go department by, some of these departments have like 5 professors and 3 students. You can look this stuff up, whatever, this isn't just me making stuff up. And other departments, think health and exercise science, biology, oh, tons of students. Just because a department exists doesn't mean it is equal to all other departments in terms of revenue or student population or whatever. Same thing with amino acids. Just because there are these 20 amino acids and you can sense a bunch of them, doesn't mean methionine is equal to leucine or arginine. There is a pathway through which methionine promotes mTOR through RAGs by inhibiting SAMTOR, which is a promoter of GATOR1, and GATOR1 is the primary inhibitor of the regulator localization of mTOR, protein activation of mTOR. Threonine sensing, there's another one over here. If we go back, you can see threonine sensing, TARS2 right there. And so if we bind threonine to TARS2, it's a promotion. Green means promotion, red means inhibition. And so with threonine, there's another promotion. A weak one, it's super weak, but it is a promoter of that RAG complex, threonine. So write that one down too. Now other amino acids matter too. The other, glutamine aside, the other amino acids are not well outlined. The effects are not very potent, very modest effects, and the mechanisms aren't that well outlined. Glutamine we'll talk about more in just a second, but activates mTOR in a RAG-dependent manner, as well as another MAPK signaling. But also alanine, histidine, serine, valine, all of these have been shown to be recognized by the cell. Linked to mTOR complex 1 activation very weakly. Valine and histidine, those are both essential amino acids. Those two are not. Those are totally non-essential. But the cell still can recognize them. But again, just because a cell recognizes that an amino acid is there doesn't mean mTOR is so responsive to it. Now getting into the glutamine effects beyond RAGs, you can see an MAPK activation with glutamine. ERK, extracellular signal regulated kinase. This study is in mice. But again, there's a lot of rodent studies, cancer cell studies, and we have to generalize for a lot of this stuff. But that's glutamine right there. An increase in extracellular signal regulated kinase. This is MAPK signaling, MAPK, RSK signaling. Within 5 minutes, pretty fast time course of this. And what you see is an intracellular calcium increase. With glutamine, you get more intracellular calcium. That intracellular calcium seems to be the signal to get that MAPK pathway activated. So glutamine has a couple of different effects. A couple of other amino acids are weakly recognized in this same way. Phenylalanine and cysteine, both of those also are recognized in a similar way that glutamine is. Very weak though. This page, again, this will be online, you'll be able to see it, but this is a nice description up here, the brown font part of MAPK activation. And you know this stuff because we've been talking about how growth factors, things like IGF, insulin-like growth factor, these growth factors, when they go through their signaling cascade, they bind to a cell surface receptor, the G protein coupled receptor, and then that signal gets transmitted into the cell through MAPK signaling. And we see the same thing with glutamine in a slightly different way. You know, there's MEK, ERK, and looking at all these different amino acids, and phenylalanine has an effect. There's glutamine has an effect, and then cysteine has a pretty good effect. Everything else is a little bit weak. Glutamine's the most potent. Phenylalanine, cysteine have a little bit of an effect. Everything else tends to be pretty weak. And it's intracellular calcium that gets released. It's not from an extracellular source, as though it's stretch-activated channels. It's not like glutamine. You eat a bunch of glutamine, and that activates your stretch-activated channels, so extracellular calcium comes in. It's intracellular calcium that gets released, and the intracellular calcium stimulates MAPK signaling. And you know, MAPK crosstalks with mTOR, converges with mTOR. So glutamine has its own effects. Glutamine has its own pathways. We can talk more about this stuff later. I want to get to the end of these slides, but this is just a general summary of protein signaling. Tends to be through the relocalization of mTOR. Now, glucose. One thing, we're going to talk a lot about AMPK and glucose and glycogen, that regulation of AMPK. There are beta subunits on AMPK that glycogen can bind to, inhibiting AMPK. So if you're carb-rich, if you're carbohydrate-rich, AMPK has an inhibition. Glycogen binds to the beta subunits on AMPK with an inhibitory pathway. We'll talk about that in the next lecture. But also, these intermediates in glycolysis have an inhibition effect. An mTOR promotion effect. And so one of them, you know, hexokinase. Hexokinase is the enzyme that works on glucose. The first step in glycolysis that works on glucose. So, amino acids, the major pathway is to RAGs. Major pathway is to RAGs to get mTOR to the work site. Protein, amino acid, we get mTOR to the work site. Once it's at the work site, then we have PI3K and MAPK can stimulate it. But they can't stimulate it if it's not at the work site. And also, there's some kind of a weak activation of things like glutamine going through MAPK, some additional activation. We'll talk a lot more about AMPK, LKB1, and AMPK. We'll talk a lot more about this stuff tomorrow, or Wednesday. And LKB1 does a lot of stuff. Liver kinase B1, just like PKB or AMPK, is phosphorylating stuff. But AMPK is the primary effect, at least the one that we care about. Let's skip some of this stuff. Now again, reactive oxygen species. Before, when I was talking about mechanical loading and reactive oxygen species share a relationship. Same thing with nutrition and chemical reactive oxygen species. They share a relationship. Each one of these categories does not live in isolation. There's overlap, signaling overlap in some of these things. But nutrition, lipids, think phosphatidic acid, prostaglandins, but also androgens, estrogens, androgens. Those would be in the fats as promoters of mTOR. Carbohydrate is a promoter of mTOR also. You can just eat carbs to get insulin, PI3K signaling. We'll talk a little bit more about carbohydrates. Proteins, mostly about GTP-loaded RAGs getting mTOR to the work site. Okay, so I'm just going to read a few slides that summarize this combined effort of protein and growth factors. So mTOR is activated by need. Need is what activates mTOR. So if you're using a cell, consider Henneman's size principle, orderly recruitment, Henneman's size principle. If you're using a cell, you may need to repair that cell. If you're using it, you're banging it up, you may need to repair it to reinforce it against future bouts of that same stress. When a cell is damaged, you get an inflammatory response, immune, chemical, inflammatory response. These chemicals function as a marker for the magnitude of damage and the corresponding necessity of repair. There's a signal for the architectural disruption, and then they initiate that repair through PKB and MAPK signaling, mostly PKB. Mechanical loads are also transmitted to demonstrate what this cell is doing, because what you're currently doing, what you're doing in this moment, is an indicator of what you'll need to do in the future. How you behave today is a good predictor of how you'll behave tomorrow and the next day. Sensing of chemicals and mechanical loads, these signals, these both will signal the necessity of repair and remodeling. They don't announce that nutrition is coming. They announce a need to repair. You've banged this thing up. Perhaps you'll have inadequate calories on board, but you still need to do some translating. You know, just like after an earthquake in Haiti or something, there may not be the funds, but you still have to fix stuff, right? So need. Nutrition is an activator of mTOR also. You don't want to grow if you lack macronutrients. Nutrition is required to grow. If you lack those macros, you don't want to start growing. When you do present a cell with an abundance of calories, that's when you turn mTOR on. It's sensitive to fed and fasted states. In a fasted state, mTOR is off. It's really hard to turn on mTOR in a fasted state. Again, despite growth hormone. It's nearly impossible to turn on mTOR in a fasted state. During a fed state, it's much easier to turn on mTOR, because you don't want to translate a bunch of proteins when you don't have protein available to translate. So it detects nutrient availability. And then the assumption of nutrition. So there are multiple ways of showing your cells that you have an abundance of calories. You can eat those calories. You can provide the sustenance. You can eat them. Or, number two, don't provide the calories, but promise they're on the way. Initiate this PI3K, PKB signaling. Insulin, you know, just like I said, evolution couldn't anticipate the separation of these different amino acids and eating leucine in isolation. Injecting insulin is a way of promising the cells, oh, glucose is on the way. Glucose is coming, go ahead and let's activate some red, right? So this assumption of nutrition, there are ways of hijacking that system. But then this combination of activators is really what's needed. You need insulin or some growth factor, and you need nutrients. Nutrients to get mTOR to the work site, there's a RAB complex, and growth factors to get REB to turn it on. REB is going to be at the work site. You have to get mTOR there through nutrients. Nutrients get mTOR there, amino acids get mTOR there, and growth factors are able to turn it on once it's there. Last slide. For like nuclear weapons, there's a 2-key system. Think Harry Potter and Gringotts, right? The Goblin Bank or whatever it is. There's a 2-key system. It's not one person can't come in and control the nukes, right? Maybe they're having a terrible day. They're on like an awful day. All they need to do is stop some guy from going to the women's bathroom. It's a very specific story. It's just like there's a terrible day, and they go in and launch the nukes. You need a 2-key system. mTOR is highly regulated, and you need the 2-key system to turn it on. You need nutrient availability and growth factors. Both of those, mTOR gets turned on, and there are ways to hijack that system, but inhibition is the default. Inhibition is the default. And so AMPK is a primary inhibitor. We'll talk about AMPK on Wednesday, and then Friday and Monday, we'll do strategies of promoting mTOR. We'll do science-based growth advice. All right, that's it. And then the test is on Wednesday. Next Wednesday, not this Wednesday. A week from Wednesday is the test. That's a big stretch.
I'll need to.